All right. Hi, everyone. Thank you all for watching. So my name is Jared Balbona, and today I'll be talking about a project on estimating vertical transmission and genetic nurture using polygenic scores. This is a project I've been working on with Matt Keller and Younggang Kim here at the University of Colorado Boulder, and it's one that I'm very excited about. So without further ado, I will start with a brief overview. As you all know, parents and offspring resemble one another in a variety of ways. One of the most commonly used examples of which, at least in our field, is that highly educated parents tend to have highly educated offspring. So there are a couple different explanations for why this similarity between parents and kids occurs. The first and perhaps most obvious of which is shared genetic effects. So the parent has a strong genetic predisposition for higher education, which she transmits to her daughter, giving her daughter that same predisposition. However, we can consider an alternative explanation in which being highly educated itself influences how parents raise their child. So, for example, a highly educated parent may be more likely to encourage a positive attitude towards schoolwork or to read to their child. All of these different behaviors that collectively could have a potential influence on the child's education level down the line. So this effect of a parental phenotype on an offspring one is called vertical transmission, and it's mediated here by what's called the offspring's familial environment. That is the environment in which the child is raised. So of course, this vertical transmission path and this shared genetic one are not mutually exclusive, but rather they co-occur, meaning that children with genes conducive to higher education are more likely to also have environments conducive to education. In other words, vertical transmission means that a person's genes and environment will covary with one another, a covariance that has recently been termed genetic nurture. As you can probably guess, this genetic nurture covariance makes it very challenging to tease apart genetic and environmental pathways from one another, which is something that we need to do in order to estimate and understand the relative importance of parenting in shaping who children become. Furthermore, if unaccounted for, genetic nurture will bias the estimates of heritability that we get from both twin-based and genome-wide studies of genetic effects. And it does so in some kind of unpredictable and counterintuitive ways, making it all the more important for us to tease apart. So to this end, researchers have begun focusing not only on the genetic variants that parents transmit to their kids, but also on the set of alleles that parents have but do not transmit to their children, shown up here. So like these transmitted alleles, the non-transmitted alleles still have a direct impact on the parental phenotype, which will in turn influence the offspring's familial environment and subsequently the offspring's phenotype. However, unlike the transmitted alleles that are shared between parents and offspring, these non-transmitted alleles do not have a direct effect on the offspring. So notice that there's no arrow right here. So thus, by essentially subtracting these paths in green from these paths in red, we can get an unbiased estimate of this direct genetic effect. That is, the actual causal effect of these alleles on the phenotype. And we can use that to, in turn, get an unbiased estimate of vertical transmission. So recently, Matt Younggang and I took this logic and instantiated it into a series of structural equation models for a paper we recently published. So this is one of those models, which contains all of the pieces I just described. Namely, it has these three Ys to represent the parent and offspring phenotypes. And for those of you who do structural equation modeling, note the circles here. So the parental phenotypes don't actually need to be observed in our models. They can be represented as latent variables if need be. So in addition to these three phenotypes, we also have these three big Fs representing each person's familial environment, that is the environment in which they were raised. We also have these NTs, two NTs, representing polygenic scores made from the non-transmitted portions of each parent's genotype, and these T's to represent polygenic scores made from the transmitted portions of the parental genotypes. So note that these T's have arrows going to both the parental and to the offspring phenotypes. So this model compares relationships between all of these different pieces to estimate parental effects, and in doing so, it builds on past work in a number of ways. Namely, using only a polygenic score it can provide unbiased estimates of heritability, vertical transmission, and genetic nurture, and it can do so even when the polygenic score being used is not very predictive, which is great considering that polygenic scores generally do explain a small proportion of trait heritability right now. Secondly, they can properly account for various types of assortative mating at both equilibrium and disequilibrium. 3, they are not in principle biased by missing data if those data are missing at random. So thus, these models do not require complete trios. If we just have father-child or mother-child pairs, that works too. You'll just need a larger sample size to achieve equivalent power. Fourth, as will hopefully become clear over the course of this demonstration, these models are very easy to use, relying on relatively simple math. And 5, which I hope will also become clear, they can allow for a vast number of extensions to best fit the trait and data being worked with. So that was a really quick overview, but moving on from there, I'll now pivot a bit and start talking about how these estimates are actually derived using OpenMX. So OpenMX is a package in R that's great for working with structural equation models, and it's one that I'm guessing a lot of you are probably already familiar with. So we'll be running a script I wrote in OpenMX on some example data that I simulated. So you can download the code and the practice data from here, tinyurl.com/SEMPGS2021. That should redirect you to my Google Drive. But if for whatever reason this nice little short link doesn't work out, you can also try to type in this link here, which will also take you to the same place where you can download the data and the script I'll be going over here. So using the simulated data, we'll be trying to answer the question, how does parental educational attainment influence offspring educational attainment? Now, as with just about any structural equation modeling, this process begins with choosing the actual model. So like we described in our paper, there are several different variations of our models, each designed to address different types of questions with different types of data, and they might entail some different assumptions along the way. So because of that, there are some questions to consider when you start thinking about the type of test you'd like to run. First, is this a trait that parents may be assorting on, and has this assortment likely been consistent across generations? So in other words, is this a quality that partners are more likely to be similar on, such as education or height? And is it possible that the importance of this trait in mate selection has changed over time? So for height, probably not. Tall people have probably always preferred tall people, short people with short people. But for education, it's very possible that it's become increasingly relevant over the past 100 years or so. Secondly, are there good existing estimates of heritability for this trait? And could the heritability be age or sex dependent? 3, how many trios and parent offspring pairs do I have available? Do they all have measured phenotypic data? 4, do I have any other nuclear family relatives like grandparents, siblings, or spouses in my data? So one thing to point out for these first 2 points, which are a little bit more conceptual, you don't need to have actual answers to those questions. These are just things to consider and are also things that you could test using our models if they're of interest. And I should also point out, this is not by any means an exhaustive list, or this is just something to consider as you're getting started. So moving back to those models, we can stick with the model that I showed earlier, this one right here, which makes 2 important assumptions. First, it assumes that there is assortative mating in both the parental and grandparental generations. So following the educational attainment example we're working with here, it assumes that in the parental generation and in the grandparental generation, spouses preferred to mate with people who are phenotypically similar to them, who have similar education levels. So secondly, this model also assumes that the polygenic score explains the full trait heritability, which of course is not true for any trait at present. So this specific model is mostly useful didactically. I wouldn't recommend using it or the example script on actual data to estimate parental effects. But we do have other models that will relax this second assumption, which will be more effective for actual analyses. So just something to point out. Okay, so as mentioned earlier, our models require five observed pieces of information. So first, the offspring's phenotype must be observed. So in this case, it's a value indicating the amount of schooling completed by each individual in the sample. We also have, our models also require 2 educational attainment polygenic scores for each offspring, one from the set of alleles they inherited from their dad, one from the set of alleles they inherited from their mom, as well as 2 polygenic scores made from the non-transmitted portions of the parental genotypes. Finally, as I touched on earlier, we can incorporate observed parental phenotypes if we'd like, which will increase our power, but they are not necessary, and for that reason, they're not included in the example script I'll be going over. So these are our observed values that we feed into OpenMX. So if I switch over to R, this is the script that's posted on Google Drive. If we read in the data here and take a look at it, you can see that the data looks exactly the same as what I showed on that PowerPoint slide. So one column for each, one column for each parameter, phenotype, and the 4 polygenic scores, 1 row for each individual. So you can see, for example, this individual has a lower polygenic score and lower phenotypic score than, say, this individual here. And all the columns for this particular script do need to be in this specific order, which is something you can adjust when you do this yourself. So once we've read in the data for this script, we'll specify a couple of options. You can play around with which options you'd like to use. Some might be better suited to specific types of tests than others. However, one that is a requirement for our models is this NP-SOL optimizer option, which I'll explain in a second. All right, so the first thing to do is to create the different variables that our models will be estimating, which we're doing here. So each of these we'll get an estimate for each of these down the line. We're explaining that they're free to be estimated. We're not constraining them to a specific value. And below that, for some of those variables, we'll be giving algebraic constraints. So these algebraic constraints are derived from path tracing. They're all described in pretty great detail in our paper, if that's of interest to you. And one important thing to note, you can see, for example, that Vy is a function of Vf, and Vf is a function of Vy. Similarly, G is a function of omega, omega is a function of G. This type of circularity is a hallmark of a lot of causal models, and it's definitely present in our models. These are called nonlinear constraints, and to be estimated, that's where this NP-SOL optimizer comes in. So NP-SOL is a software package that OpenMX implements to estimate nonlinear constraints, and it does so in a way that's really efficient and effective and works really well for these kinds of analyses. So once we create the variables and provide algebraic constraints, we'll also provide some equality constraints. In this case, just saying that these variables are equal in value to the algebra here. So nothing super interesting in this case, but for other more complicated tests that you might want to run, you can play around with the equality constraints here. You can say things like, I want the genetic values to be constrained for parents and for their offspring. Or you can relax that assumption and say that you don't want the parental and offspring's genetic values to be the same. Or you can do that for assortment, for parental effects, whatever you want. As with everything in the script, you can play around with different options to get different results. So once we've specified all the different variables, we will then provide an expected covariance matrix here. So the idea behind that, and I'll go back to PowerPoint for this, is that OpenMX will take all of the different variables that are inside of the expected covariance matrix and simultaneously get estimates for each one in a manner that minimizes the distance between the estimates and the observed values as much as possible. So it'll try to come up, say, with values of delta, G, W, and K that approximate 0.28 as closely as possible, for example, and it'll do that for all of these. So moving back to the R script, specify all these options. And if I don't get to anything, hopefully the annotations will describe what's going on, but if you have any questions, feel free to reach out to me. So here you can see we have a couple of nested for loops. These are trying out different combinations of start values for delta, V, E, and F. And if we run that. So using these different start values, OpenMX will iteratively test out different combinations of parameter estimates in a way that, like I said, minimizes the distance between the expected and the observed covariance matrices. So once we run that, we get a couple of warning messages here. And fortunately, these don't look like anything to really worry too much about. Most are saying that the start values are not feasible, which is to be expected if we're telling OpenMX that phenotypic variance is very heavily dependent on genetic factors, and that it's also very heavily dependent on environmental factors. It's possible that those two things can't coexist at the same time, and in which case it will produce this warning and NAs in your data, which is totally fine. So once we've tested out all those different combinations of start values, this final step, step 4, we'll select the one with the lowest log likelihood and produce it at the bottom in a data frame called results summary, shown here. So from results summary, this is really what the whole script is all about. We can see the phenotypic variance and the relative amounts of variance due to additive genetics, parental effects, and unique environment. And I also have a key here at the bottom to explain what these different columns mean if any of you forget. So in these results, we can see that parental effects are explaining a relatively small proportion of the phenotypic variance, especially compared to additive genetics and unique environment. So if these results are to be believed, it appears that parents have relatively little influence on the amount of schooling that their children choose to pursue later in life. Of course, it's possible that if you try a different model that makes different assumptions, tests different constraints, it may fit even better than this one, and you might see a slight change in the relative importance of genetics and environment. So that's something to consider. And also, I know, not to belabor the point, I know I've said this a couple times, but again, these are simulated data. They're totally made up. Actual empirical evidence suggests that parents do have a relatively substantial influence on their offspring's education level. So take these numbers with a big grain of salt. Okay, so that is the script. And moving back to the PowerPoint, I can close now by talking about a couple of the other models we built and the types of questions that they can address. So importantly, a lot of these models are part of upcoming publications in our lab. So they haven't been posted on our GitHub just yet, but hopefully they will very soon. So first, we have models that can leverage sporadic relatedness in large samples. So you can derive estimates of parental effects without needing to actually recruit a large number of trios. So for example, there aren't a ton of trios in the UK Biobank, but there are a large number of siblings and spouses. So you can use those to derive estimates, which is a lot easier than getting a hold of actual trio data. 2, these models can be used bivariately without complicating the math too much. So we can test the effect of a parental trait on a different offspring trait to get an unbiased estimate, which has never been done before. We can also model various types of assortative mating, like primary phenotypic assortment, genetic homogamy, or social homogamy. 4, we can test whether effects are sex dependent. So this can be used to address questions like, do fathers have a greater influence on offspring substance use than mothers? Are these effects dependent on whether they have a son or a daughter? Things like that. And similarly, we can examine how effects have changed across previous generations. So you can look at how mate preferences have changed across generations, like I said earlier, but you can also look at whether the influence of parenting has increased or decreased over time. So maybe peer influence becomes more important for certain traits, or maybe parenting remains equally important across time. So a lot of different avenues for future studies, a lot of things that we're really excited about, and hopefully we'll start to release data on in the upcoming future. So with that, I'd like to thank my co-authors, Matt and Yanggong, and all of you for listening. I know that was a lot of information in a pretty short period of time, so feel free to email us if you have any questions. We're happy to help out, to help try to troubleshoot anything we can do to help. So thank you all very much.